There is a very good chance that right now, sitting somewhere in your kitchen, growing wild in a field near you, or sold for less than $2 at a market you pass every week, is a plant that clinical researchers have spent years studying for its effects on inflammation, infection, cognitive decline, and metabolic disease. Not a supplement. Not a patented compound. A plant. And here is what is strange about this. We are living in an era of pharmaceutical breakthroughs, precision medicine, and billion-dollar drug development pipelines. Yet some of the most consistent findings in modern biomedical literature keep circling back to the same organisms that healers in ancient Egypt, traditional Chinese medicine, and Ayurvedic practice cataloged thousands of years before a single clinical trial ever existed. That is not coincidence. That is convergence. Today, we are walking through 10 of them. Plants with documented pharmacological mechanisms, real peer-reviewed research, and a history of therapeutic use so long that it predates writing itself. Stay with me through the entire list, because number 4 has a mechanism that researchers did not fully understand until the early 2000s, and it changes how you will think about pain. And number 2, most people dismiss without a second thought. The science behind it is anything but dismissible. Let us get into it. Plant number 10, Ashwagandha. The word adaptogen gets thrown around loosely in wellness circles. It has become one of those labels that sounds scientific but gets attached to anything that makes people feel slightly better on a Tuesday afternoon. So let us be precise about what an adaptogen actually is, and why ashwagandha earned that classification before the word even existed. Withania somnifera has been a cornerstone of Ayurvedic medicine for over 3,000 years. Ancient practitioners used it for what they called rasayana, a term that translates roughly to rejuvenation. The root, specifically, was prepared as a powder mixed with warm milk, given to people recovering from illness, to warriors before long campaigns, to the elderly whose strength was failing. The application was not random. It was based on thousands of years of observed outcomes. Modern pharmacology has now identified the active compounds responsible. Withanolides, a class of steroidal lactones unique to this plant, appear to modulate the hypothalamic pituitary adrenal axis. That is the system that governs your cortisol response. When this axis is dysregulated, which is increasingly common in populations with chronic psychological and physiological stress, the downstream effects touch everything. Sleep quality, immune function, reproductive hormones, cardiovascular load, inflammatory markers. All of it runs through this system. Multiple randomized controlled trials have confirmed that standardized ashwagandha root extract produces statistically significant reductions in serum cortisol. One double-blind study published in the Indian Journal of Psychological Medicine found that participants taking 300 milligrams of root extract twice daily over 60 days showed a cortisol reduction of approximately 27% compared to placebo. That is not a marginal finding. The thyroid research is equally notable. Studies in patients with subclinical hypothyroidism have shown improvements in TSH and T4 levels following supplementation. The mechanism appears to involve stimulation of thyroid hormone synthesis rather than merely masking symptoms. And for people dealing with exercise-related fatigue and recovery, controlled trials have demonstrated improvements in VO2 max, muscle strength, and time to exhaustion. Ancient practitioners did not have the language for any of this, but they were observing the same outcomes, generation after generation, and building a pharmacopoeia around what they saw. The word adaptogen was coined by Soviet scientist Nikolai Lazarev in 1947. Ashwagandha had been filling that role for 30 centuries before he named it. Plant number 9, elderberry. Most people encounter elderberry in the form of a gummy vitamin at a pharmacy checkout. It sits alongside the zinc lozenges and the vitamin C packets, and it gets treated with the same mild skepticism that most people extend to anything sold in a chewable bear shape. That skepticism is understandable. It is also worth examining. Sambucus nigra, the black elderberry, has one of the most consistent bodies of clinical research of any plant on this list when it comes to acute respiratory illness. Multiple randomized controlled trials have demonstrated that standardized elderberry extract can reduce the duration of influenza illness. A meta-analysis published in Complementary Medicine Research analyzed data from multiple studies and found a substantial reduction in flu duration among participants who took elderberry extract compared to placebo groups. The mechanism is pharmacologically interesting. Elderberries contain a high concentration of anthocyanins, specifically cyanidin-3, glucoside and cyanidin-3, sambubioside. These compounds have demonstrated direct antiviral activity in laboratory settings, appearing to bind to viral particles and inhibit their ability to penetrate host cells. Additionally, elderberry activates cytokine production in ways that prime the innate immune response. The immune activation component is real, documented, and measurable. Traditional use across European folk medicine was extensive. The elder tree was considered so medicinally significant that it was planted near homes specifically as a health resource. The flowers were used for fever and inflammation. The berries for viral illness and respiratory conditions. The bark, though toxic in its raw form, was used in carefully prepared formulations for joint pain. Every part of the plant had a documented application, and modern research has now worked backward from those applications to identify why. One caution worth stating: raw elderberries contain sambunigrin, a cyanogenic glycoside that can cause nausea and vomiting if consumed in quantity. Cooking or standardized extraction eliminates this compound. The elderberry in reliable commercial preparations is processed specifically to remove it. This is not a reason to avoid elderberry. It is a reason to understand what you are taking and where it comes from. Plant number 8, holy basil. There is a plant in the mint family that is considered so sacred in Hinduism that it is found growing in a pot at the entrance of millions of homes across South Asia. Not as a decoration. As a daily ritual. Leaves are added to water, to tea, to food, and offered in prayer, and this practice has continued, uninterrupted, for somewhere between 3,000 and 5,000 years. Ocimum tenuiflorum. Holy basil. Tulsi. The reverence is not arbitrary. It is pharmacologically grounded. Holy basil contains a dense array of phytochemicals, including eugenol, rosmarinic acid, ursolic acid, and multiple flavonoids. The combination creates what researchers describe as a broad-spectrum adaptogenic and anti-inflammatory profile. But what distinguishes holy basil from many other adaptogens is the specificity of its effect on blood glucose regulation. Multiple human clinical trials have now demonstrated that holy basil supplementation produces statistically significant reductions in fasting blood glucose, postprandial blood glucose, and HbA1c in people with type 2 diabetes. One study published in the Journal of Clinical and Diagnostic Research showed that 2.5 grams of holy basil leaf powder taken daily for 3 months reduced fasting blood glucose by over 17% and postprandial glucose by over 7% compared to control. The mechanism appears to be multifactorial. Eugenol has demonstrated beta cell protective effects in pancreatic tissue. Ursolic acid has shown insulin-sensitizing properties. And the adaptogenic compounds modulate the cortisol-driven glucose dysregulation that makes stress such a significant factor in metabolic disease. Beyond blood sugar, holy basil has demonstrated antibacterial activity against a range of pathogens including Staphylococcus aureus and Escherichia coli. Its anti-inflammatory effects, particularly the inhibition of cyclooxygenase enzymes, Place it in a pharmacological neighborhood usually occupied by nonsteroidal anti-inflammatory drugs, without the gastrointestinal side effects that make long-term NSAID use problematic. The people who planted Tulsi at their doorways were not performing empty ritual. They were maintaining a daily pharmaceutical practice that took millennia of careful observation to build, and that modern biochemistry is still in the process of fully mapping. Plant number 7. Milk thistle. The liver does not announce when it is struggling. It does not produce pain the way an injured muscle or a damaged joint does. The liver compensates silently, redirecting function, working around damage, until the point at which it cannot. And by the time most people discover a liver problem, the progression has often been underway for years. This makes hepatoprotective compounds particularly valuable. And among them, one plant stands out with unusual consistency across both traditional use and modern clinical evidence. Silybum merianum. Milk thistle. The active compound is a flavonolignin complex called silymarin, concentrated in the seeds of the plant. Silymarin has been studied for its effects on liver tissue for over 40 years, and the research is substantive enough that intravenous silymarin is used in European emergency medicine as a treatment for Amanita phalloides mushroom poisoning, one of the most acute and lethal forms of liver toxicity known. The mechanisms are multiple. Silymarin appears to stabilize hepatocyte membranes, reducing the permeability that allows toxins to cause cellular damage. It inhibits the uptake of toxic substances into liver cells by competing for the same transport proteins. It stimulates protein synthesis in hepatic cells, accelerating the regeneration of damaged liver tissue. And it demonstrates antioxidant activity specifically within liver cells, scavenging the free radicals that drive inflammatory damage. Clinical trials in patients with non-alcoholic fatty liver disease, alcoholic liver disease, and drug-induced hepatotoxicity have consistently shown improvements in liver enzyme markers, including ALT and AST, following silymarin supplementation. The effect size varies, and silymarin is not a substitute for addressing the underlying cause of liver stress, but the hepatoprotective activity is real and reproducible. Traditional European herbalism used milk thistle seed preparations for liver and gallbladder complaints for centuries. What is remarkable is not just that this use was validated, but the precision with which the traditional application anticipated the biochemistry. They did not know what a hepatocyte was. They knew that this plant helped people whose livers were failing, and they were correct. Plant number 6, ginger. If you had to identify one plant on this list that bridges the largest gap between its reputation and its actual pharmacological power, ginger would be a serious candidate. Most people think of ginger as something that settles a mildly upset stomach. An ingredient in tea when you have a cold. A flavor in holiday baking. This is like describing aspirin as something you take when your head feels a bit heavy. Zingiber contains two classes of compounds that are among the most studied natural anti-inflammatories in biomedical literature. Gingerols, which are abundant in fresh root, and shogales, which form when ginger is dried or heated. Both classes inhibit prostaglandin synthesis by blocking cyclooxygenase enzymes. That is the same pathway targeted by ibuprofen. That is not a metaphor or an approximation. It is the same biochemical mechanism. But ginger does something ibuprofen does not. It also inhibits leukotriene synthesis by blocking the enzyme 5-lipoxygenase. Leukotrienes drive a different branch of the inflammatory cascade, one that COX inhibitors do not touch. This dual pathway inhibition gives ginger an anti-inflammatory reach that single-mechanism pharmaceuticals cannot match. Clinical research has confirmed this across multiple conditions. A randomized controlled trial in patients with osteoarthritis of the knee found that standardized ginger extract produced significant reductions in pain and functional disability. Studies in dysmenorrhea, where inflammatory prostaglandins are the primary driver of pain, have shown ginger extract performing comparably to ibuprofen in reducing pain severity. Research in chemotherapy-induced nausea has consistently shown efficacy, and ginger is now commonly recommended in oncology support protocols for exactly this reason. The antiemetic effect operates through a different mechanism entirely. Gingerols and shogales appear to act on 5-HT3 receptors in the gastrointestinal tract, the same receptor targeted by pharmaceutical antiemetics. They also modulate gastric emptying and reduce the hyperreactivity of smooth muscle that produces nausea. Ginger has been used medicinally for over 5,000 years across South Asian, East Asian, and Middle Eastern traditions. For much of that history, it was treated as medicine first and food second. We inverted that relationship somewhere along the way. The biochemistry has not changed. If you want to keep learning about what nature has quietly kept available for your body, this is exactly the kind of content we make. Hit subscribe and turn on notifications so you do not miss what is coming next. Plant number 5, Echinacea. The immune system is not a single thing. It is a layered, coordinated response system that involves physical barriers, innate immunity, adaptive immunity, cellular responses, and humoral responses, all operating simultaneously and influencing each other in ways that researchers are still working to fully characterize. Plants that modulate immune function do not do it by simply boosting everything. That framing, which you see in marketing copy constantly, is not how immunology works. Meaningful immune modulation means upregulating specific responses, calibrating inflammatory signaling, and supporting the infrastructure that allows immune cells to do their jobs efficiently. This is where Echinacea becomes genuinely interesting. There are multiple species used medicinally, primarily Echinacea purpurea, Echinacea angustifolia, and Echinacea pallida. The pharmacological landscape is complex because different parts of the plant and different species have somewhat different active compound profiles. The primary active constituents include alkylamides, polysaccharides, caffeic acid derivatives including echinacoside and succoric acid, and glycoproteins. The research on Echinacea and upper respiratory tract infections has been contested, which is worth acknowledging. Meta-analyses have produced mixed results, largely because different studies use different preparations, different species, different plant parts, and different doses. When research isolates standardized, whole plant preparations of Echinacea purpurea in particular, the signal becomes considerably more consistent. Multiple trials have shown reductions in both the incidence and duration of common cold infections in people who took standardized preparations prophylactically. The more compelling and less debated research involves Echinacea's effect on macrophage activation. Laboratory studies have consistently demonstrated that Echinacea polysaccharides activate macrophages, increasing their phagocytic activity and their production of cytokines that coordinate downstream immune responses. The alkylamides in Echinacea angustifolia and purpurea have shown specific binding affinity for CB2 receptors in the endocannabinoid system, which are expressed on immune cells and play a role in regulating inflammatory responses. Native American medicinal traditions used Echinacea extensively for wound treatment, snake bite, toothache, and infection for centuries before European contact. European settlers adopted it rapidly in the late 19th century, and by 1900 it was the most widely used medicinal plant in North America. The interest never disappeared. It simply became entangled in regulatory debates and quality control problems that made the research messier than the plant's actual pharmacology deserves. Plant number 4. Turmeric. In 1995, a paper published in the Journal of Natural Products described a compound that inhibited nuclear factor kappa B, a transcription factor that researchers had recently identified as one of the master regulators of inflammatory gene expression. Inhibiting NF-κB means reducing the production of a cascade of inflammatory mediators simultaneously. It was the kind of finding that generates immediate scientific attention. The compound was curcumin. The source was a root that Indian and Southeast Asian traditional medicine had been using for over 4,000 years. Curcumalanga. Turmeric. The research that followed curcumin into mainstream biomedical literature has been extensive to the point of being difficult to summarize briefly. Over 10,000 papers have been published on curcumin's effects. The challenge with curcumin is not whether it has pharmacological activity. It does. The challenge is bioavailability. Curcumin is poorly absorbed in standard form, metabolized rapidly, and eliminated quickly. This is why much of the early clinical research showed inconsistent results. The compound works. Getting it to relevant tissue concentrations in the human body is the problem. Modern formulation science has addressed this. Curcumin combined with piperine, the active compound in black pepper, shows bioavailability increases of up to 2,000%. Lipid-based formulations, nanoparticle delivery systems, and phospholipid complexes have all demonstrated significant improvements in absorption and tissue distribution. Once bioavailability is accounted for, the clinical evidence becomes considerably more compelling. Trials in rheumatoid arthritis have shown curcumin performing comparably to diclofenac, a pharmaceutical anti-inflammatory, without the gastrointestinal toxicity. Studies in inflammatory bowel conditions have shown reductions in disease activity markers. Research in metabolic syndrome, cardiovascular risk factors, and neuroinflammation is accumulating. The NF-κB inhibition story is only the beginning. Curcumin also modulates the activity of COX-2, the enzyme that produces pro-inflammatory prostaglandins. It inhibits lipoxygenase enzymes. It downregulates certain cytokines. It appears to influence multiple nodes in the inflammatory signaling network simultaneously, which is why researchers have described it as a pleiotropic agent, something that acts through many pathways rather than one. Ancient Ayurvedic formulations combine turmeric with black pepper and fat as a matter of standard practice. This was not coincidental. The pharmacological rationale had been worked out empirically across thousands of years of clinical observation, long before anyone had a language for bioavailability enhancement. Plant number 3, Valerian. Sleep is not a passive process. It is an active neurological state that the brain coordinates through multiple neurotransmitter systems, hormonal rhythms, and cellular signaling cascades. When this coordination is disrupted, the consequences are not limited to tiredness. Chronic sleep disruption has been linked to metabolic dysregulation, cardiovascular risk, immune suppression, neurodegeneration, and significant psychiatric morbidity, which makes plants with genuine, documented effects on sleep architecture worth understanding carefully. Valeriana officinalis has been used as a sedative for at least 2,000 years. Ancient Greek and Roman physicians, including Galen, prescribed it for insomnia, anxiety, and nervous tension. Medieval European herbalism used it so extensively that the plant was cultivated in monastery gardens specifically for medicinal purposes. It remained in official pharmacopoeias in multiple countries through the 19th and into the 20th century, when it was gradually displaced by synthetic benzodiazepines. The primary mechanism involves gamma-aminobutyric acid, GABA. This is the brain's principal inhibitory neurotransmitter, and it is the same system targeted by pharmaceutical anxiolytics and sedatives. Valerian root contains compounds that appear to act as GABA receptor agonists, and also to inhibit the reuptake and enzymatic breakdown of GABA, effectively increasing its availability at receptor sites. Isovaleric acid, valerianic acid, and a range of iridoid compounds called valipotriates are the primary active constituents. Clinical evidence includes randomized controlled trials demonstrating improvements in subjective sleep quality, reductions in sleep latency, and reductions in nighttime awakening. A meta-analysis in the American Journal of Medicine reviewed data from 16 studies and found that valerian may improve sleep quality without producing side effects. The results are not universally consistent, partly because valerian preparations vary significantly in composition, and partly because the active compounds are volatile and degrade in poor storage conditions. Crucially, valerian does not appear to cause the dependency, withdrawal effects, or daytime cognitive impairment associated with pharmaceutical sleep aids. The mechanism is modulating an existing system rather than suppressing it. This is a meaningful difference. 2,000 years of continuous use for the same indication, followed by pharmacological validation of the mechanism, followed by clinical trials confirming the effect. The trajectory of the research is exactly what you would expect from something that actually works. Plant number 2, Moringa. There is a tree that grows in tropical and subtropical regions across Africa and Asia that local communities have been using as both food and medicine for thousands of years. It is fast-growing, drought-resistant, and nutritionally extraordinary. Every edible part, the leaves, seeds, pods, flowers, and roots, has documented medicinal properties. And in most Western countries, it is almost entirely unknown. Moringa oleifera. The nutritional profile of dried Moringa leaf alone is striking. Gram for gram, it contains more protein than most plant sources, with a complete amino acid profile. It contains beta-carotene, vitamin C, vitamin E, calcium, potassium, iron, and zinc in concentrations that, when standardized for the amount of actual food consumed, compare favorably with commonly recommended dietary sources of each. But the medicinal research goes considerably further than nutrition. Moringa leaves contain isothiocyanates, specifically moringin and its derivatives, which have demonstrated anti-inflammatory activity through inhibition of NF-κB signaling. Multiple animal studies and a smaller number of human trials have shown Moringa extract producing reductions in inflammatory markers including C-reactive protein, interleukin-6, and tumor necrosis factor alpha. The blood glucose research is particularly substantive. Multiple studies, including randomized controlled trials in populations with type 2 diabetes, have shown significant reductions in fasting blood glucose following Moringa leaf supplementation. Proposed mechanisms include the isothiocyanates' role in improving insulin sensitivity, and the presence of compounds that appear to slow carbohydrate absorption in the gastrointestinal tract. Moringa seed oil has a lipid profile similar to olive oil, with high oleic acid content and documented antioxidant stability. Traditionally used for cooking and skin care, it has also shown antimicrobial activity against a range of bacterial and fungal pathogens. The seeds themselves have been used for centuries in water purification, and research has confirmed that Moringa seed extract can effectively reduce turbidity and bacterial load in water, a property that has practical public health applications in regions without reliable water treatment infrastructure. In parts of East Africa, South Asia, and the Philippines, Moringa is called the miracle tree. This language sounds like marketing. The biochemistry makes it sound more like understatement. If you found this valuable, subscribe right now and leave a comment below telling us which plant surprised you most. We release content like this regularly, and this is exactly the kind of thing we cover. Plant number 1, Andrographis. Number 1 on this list is a plant that most people in the Western world have never heard of. In traditional Chinese medicine, it is called Chuan Xen Lian, which translates approximately to thread through heart lotus. In Ayurvedic practice, it is known as kalme. In Southeast Asian traditional medicine, it is so widely used that it has been called the king of bitters, and also, more directly, the cold and fever plant. Andrographis paniculata. The primary active compounds are diterpenoid lactones, collectively called andrographolides. These molecules have demonstrated pharmacological activity across a remarkable range of mechanisms, and the clinical research, particularly for acute upper respiratory infection, is among the most consistent of any plant in this category. Multiple randomized controlled trials have examined Andrographis for the treatment of acute upper respiratory tract infections. A systematic review and meta-analysis published in the journal Phytomedicine analyzed data from 33 controlled trials and found that Andrographis preparations significantly reduced the severity and duration of respiratory illness compared to placebo, and in some comparisons, performed comparably to standard pharmaceutical treatments. The mechanisms are layered. Andrographolide inhibits NF-κB signaling, reducing the production of pro-inflammatory cytokines. It has demonstrated direct antiviral activity against influenza viruses in laboratory research, appearing to interfere with viral replication. It stimulates immune cell activity, particularly natural killer cells and cytotoxic T lymphocytes. And it has demonstrated antipyretic activity, reducing fever through mechanisms distinct from those of pharmaceutical antipyretics. Beyond respiratory illness, research has examined Andrographis in the context of inflammatory bowel disease. A controlled trial published in Alimentary Pharmacology and Therapeutics found that andrographolide treatment produced clinical response rates in patients with ulcerative colitis comparable to those achieved with mesalazine, a standard pharmaceutical treatment. That is a serious finding from a serious journal, and it has received far less attention than it deserves. Anti-cancer properties have been investigated in laboratory and animal research, with andrographolide demonstrating pro-apoptotic activity in multiple cancer cell lines and anti-angiogenic effects that limit tumor blood supply development. Human trials in this area are limited, and this research should not be interpreted as evidence that Andrographis treats cancer. But the mechanistic findings are generating genuine scientific interest. The traditional name, King of Bitters, refers to the extraordinary bitterness of the plant, which is a sensory signal of its dense alkaloid and diterpenoid content. Healers who named it understood intuitively that the intensity of the taste was connected to the intensity of the effect. They were not wrong. We have walked through 10 plants with documented mechanisms, clinical evidence, and histories of therapeutic use that span centuries or millennia. Ashwagandha and its cortisol modulation. Elderberry and its antiviral anthocyanins. Holy basil and blood glucose regulation. Milk thistle and hepatoprotection. Ginger and its dual pathway anti-inflammatory action. Echinacea and immune system calibration. Turmeric and the NF-κB story. Valerian and the GABA pathway. Moringa and the nutritional and anti-inflammatory depth of a plant most people have never touched. And Andrographis, perhaps the most pharmacologically compelling plant on the list, and among the least known outside of the cultures that have relied on it for generations. Here is what connects all of them. None of them are mysteries. None of them are folklore waiting to be debunked. They are organisms that evolved complex chemical arsenals over millions of years of ecological pressure, and that human beings, through thousands of years of careful observation, learned to use with remarkable precision. Modern biochemistry arrived late to a conversation that traditional medicine had been having for a very long time. The plants have not changed. The research is simply catching up.